you gonna?
Yep.
Hi, my name is Andrew Pranger. Today I'm going to give my expert to novice presentation for Purdue's COM 217, but my presentation is actually an introduction on cryo-electron microscopy and tomography. So before you listen to this presentation, you probably want to hear why you should listen to me. I am an undergraduate student with award-winning research in the cryo-electron tomography field. Additionally, I've worked very closely with my professor, Dr. Metzkes, who has gone on to get her PhD and complete multiple postdocs in the field of cryo-electron tomography. So I would hope that I'm a little bit of a respectable source for the subject matter, but I want to break into what we're actually going to talk about today. So first, we're going to determine the basic functions of cryo-electron microscopy and tomography. We're going to talk about how these work, and then we're going to talk about the practicality of it. So why does it matter to anybody in the room? So, general overview of cryo-EM. It was named 2015 Nature Methods Method of the Year, and Nature is widely considered one of the most reputable scientific journals out there, so you would think this has got to be pretty good. If that's not enough, it went on in 2017 to be the award-winning method of the Nobel Prize in Chemistry, where it won for high resolution in cryo-EM. So that being said, why is this winning so many awards? And the answer is that high resolution point. So cryo-EM has been found over the last number of years to push into subatomic and near atomic resolutions. So what that means is you have a molecular structure, so you've got atoms all connected to each other, right? And then we have how close we can actually see to the atoms. So we get what's called resolution, which if it's bad resolution, you get this giant blob. And if it's really good resolution, we can see the actual structure of the protein or what we're looking at in general. So a better display of what that looks like is this here. So we have our actual structure on the inside, and then this blue netting around it is called resolution. So over here, you can see since 2003 and up to 2019, past 2019, these have gone down even lower. We have our average reported resolutions and the best in the field. So we've gotten pretty close to zero, which would just be seeing these molecules, and that would be a really good goal to hit. That being said, so why is cryo-EM and cryo-ET so good, right? There has to be other ways of doing this. So first we have NMR, which is a form of spectroscopy where you basically take a molecule and you separate its parts based on its charges. You can recognize the structure of something based on that. It doesn't give you a very good visual image, but it gives you a lot of data. Then we also have types of crystallography. So that was how we found the structure of DNA, thanks to Rosalind Franklin. Essentially, you have to crystallize a lattice structure of your protein and pass X-rays through it, and then the X-rays create an image. So that's really hard because crystals are hard to prepare. There's other methods as well, but that's kind of the front runners in the field. So there's not a lot of really good ways to do this. So that being said, if EM is so good and ET as well, then why does it only make up 2.5% of the field? And the answer is cost. It takes about $5 to $7 million to buy one electron microscope, and you also have to get the people who are trained to run that electron microscope. So it's really hard to get into, and not everybody has access to that. But that being said, that's enough general information about it. So how does this actually work? So what we want to do is determine an isolated molecular structure of some specimen. So you might remember that one guy I had over here with the blue netting around it. That's what we want to find. We want to find the actual structure of something. That being said, we also don't need crystals. So in crystallography, you have to have crystals. In this, we don't. We want to reconstruct the data in a really high resolution so we can actually see what we're looking at and make decisions based on that. So a very quick overview of what all this is. You prepare a sample, we take that sample, we put it on a grid in the electron microscope. Electrons are passed through, so really small, high energy balls are bounced off the sample, then they produce a shadow down here. The shadow is detected, and then we interpret the data from there. And that's how we get our really cool structures. Pretty simple. Prepare a grid, scan the grid, then look at the grid with the camera, and then process your data from the camera. It's not that bad once you break it down into smaller steps. Cryo-ET, so I mentioned tomography versus microscopy. So the difference is that in ET, it's a stable field, and then we pass the electrons directly down, and that's called single particle. In Cryo-ET, we actually move the stage so you can look at the sample from different perspectives. So there's practical uses of cryo-EM and ET. So I've told you what it is. Now, why does this matter? So we have gene expression, protein synthesis, membrane proteins, viruses, neurodegenerative disease-related proteins, and immune system proteins can all be looked at by isolating them and scanning them via electron microscopy and tomography. So a first example would be viruses. So the COVID-19 vaccine, for example, what they were able to do is they were able to scan this in an electron microscope. They were able to make a 3D rendering of it, and this might look familiar to you. This is a spike protein from COVID-19. So everybody was talking about the spike proteins when they were making the vaccines. Spike protein. Additionally, we also have neurodegenerative diseases. So Alzheimer's, for example, tears away at the neurosystem. So what they were able to do here is they were able to look at the fibrils of Alzheimer's as taken from this part of the brain over here and reconstruct them and compare them between non-Alzheimer's patients and Alzheimer's patients. So, got my sources that I used for this. If you have any questions, please ask now.
That was very good.
The audience is here.